Thank you, Ravi. I'm also selectively colorblind to these different sheets. A very good afternoon to everyone. Thanks to Shah and Sharif, Dr. Nagy, and Ravi for putting together this show. Well done and kudos to everyone. We are entering the fourth revolution in agriculture. We have gone through three different revolutions. About 120 years ago, when James Watt he introduced and invented the steam engine, we have gone through a mechanization process, which leads to industrial revolution, which is followed by the green revolution. So development of novel new breeding for different crops and different plant-related trees, followed by genetic engineering. Genetic engineering is also making strides and advancements continuously, even in this new century. For the past 10 years, we have been working and looking at precision livestock farming or precision agriculture. So the missing link in terms of animal health diagnostics is bringing down the time to results. So whenever an epidemic or a pandemic breakout happens, it takes sometimes days to assess what type of strain is causing that particular breakout, what is the source, whether it is a high pathogen versus low pathogen, irrespective of the animals. So sometimes when the farm or the barn is far away from testing facilities such as our animal health laboratory, the time to results is crucial in terms of making decisions to understand and also to prevent further breakout and the disease-related issues. So the trends of future is, can we bring down the time to results? Can we move the lab from a faraway facility to a handheld tiny little chip in the barn, in the farm itself? So that is the goal behind one of the objectives of our bio-nano laboratory within the School of Engineering. Again, from an engineering perspective, high pathogen doesn't really occur all the time, probably once in a decade or so, but low pathogen does occur quite frequently. So there is a dire need from a farmer's perspective in assessing towards making strategies to manage the health of the birds and also other related animals by understanding the type of strain, discriminating and distinguishing what particular strain is actually happening in that particular moment. There are issues surrounding vaccines too. I think one of the previous speakers identified, by the time a new vaccine is being introduced, maybe another strain is actually causing. Sometimes it takes about a week for the vaccine to show its effect in terms of reducing the actual disease processes and mechanisms within the body of the bird. So considering that, one of the complementary as well as a way of controlling the disease breakout is predicting and preventing the diseases through a proactive approach. So can we be able to predict even before the disease is going to happen through a use of novel invention such as identification of biomarkers and other related environmental parameters. So it is not just one cue, one particular parameter, but putting together a multi-cue, multiplexing, parallel approach of identifying and taking into account the environmental as well as the biomarker of the bird towards identifying and predicting the disease. So from our lab, we have developed two different sensing mechanisms. So to simply put, this is a simple optofluidic technique. It is like when your grandmother checks whether there is a sugar present in her, it's a home-based, sugar-based test. So basically, prick a finger, take a droplet of blood, and stick it in a little cartridge reader, and that shows whether the particular biomarker is present or not. Same concept, same model, but the mechanism is different. Here we are using antibodies and enzymes to detect and identify the disease. So this is an optofluidic technique, optofluidic, optical, it's a light-based technique. So the reader will shine a laser at specific wavelength, and depending on the antibody specificity, it would be able to discriminate between high pathogen versus low pathogen using a special patented technology called quenching mechanism. So I was walking from the School of Engineering here to Arboretum. I feel thirsty. I drink a sip of water and I quench my thirst. Similar concept, the number of actual virus strain in a known volume of blood when it binds to specific antibody in the presence of multiple strain-related blood solution quenches specific to specific strain. So depending on the difference in quenching, we can actually identify which particular strain is present, whether it's a high pathogen or low pathogen, and so on. So we have went ahead and demonstrated 2 different strains. We also went ahead mainly towards the field deployable and portability nature. The goal is bringing the lab from the building to the hands of the farmer, a lab on a chip in an electrosensitive, electrochemical based mechanism. So we went ahead and developed another mechanism using ketosine and other protein related mechanism to be able to differentiate these strains. Main reason behind identifying and developing electrochemical versus a optofluidic is the ability to transmit the data in a real-time fashion. So as soon as we know what particular disease is happening, we need to interface to a smartphone appearing device and send the data to food safety inspectors and veterinarians. So electrochemical approaches are perceived to be better than optofluidic technique. So we went ahead and also built a handheld appearing device. So again, this is an ongoing work. Further validation are needed with real-time samples. So we're looking for collaborations from veterinarians from the Ontario Veterinary Colleges and other eminent scientists such as Dr. Nagy. So the end vision is that if there is a breakout that's going to happen, or even as a routine clinical screening tool, one would be able to just take a droplet of swab from the throat or even a litter sample from the environment of the poultry barn, mix it in a little vial, add a droplet in a little cartridge, wait for probably a minute, stick it in a reader such as this, and then wait for maybe a minute. It is going to give you what type of strain is present, and also you can actually quantify how much it is present in known volume. So the ability to quantify as well as qualitatively assess is the end vision of this particular technology. So there are a variety of economic benefits. Canadian Food Inspection Agency and World Organization for Animal Health, OIE, both are very much interested in these type of technologies because they will definitely enhance the biosecurity protocols, and there is a real need for demand for novel diagnostic technologies that will bring down the time to identify the diseases, bring down the time to results. There are also welfare-related advantages. For example, conventional techniques, when you ship samples, you have to draw vials of blood from the bird. Here we are talking about just a droplet. Prick it, a droplet would give you more than 2 or 3 different types of strains. So welfare is also associated towards introduction of technology. Again, we are not looking at replacing the veterinarians, but empowering the veterinarians and also enhancing the disease diagnostics by rapidly bringing down the detection time. So on that note, we have been working, we have also patented 2 other technologies in dairy-related area. Metabolic diseases and ketosis for dairy farms. So the Business Development Office, Tech Transfer Office of University is actually negotiating with some of the bigger players to license this technology. So in terms of 4th revolution in agriculture, here in Ontario, there is a bigger role we could play as a group. This is the end vision of the innovative detection and advanced diagnostic technologies for farmed animal health management. So not only detection of biomarkers, when we put together detection of environmental variables such as amount of ammonia that's being produced, other foul, odor, volatile metabolites, change in humidity and temperature, all these together can be put together in the form of a Internet of Things or IoT technology to be able to rapidly disseminate disease-related incidental measurement using these technologies. It's a cartoon showing 3 different sectors here. We have a poultry in the right corner, swine as well as dairy. So a simple smartphone appearing device would be able to not only detect but also in a real-time fashion has the potential to disseminate the information to the food safety inspectors, veterinarians and animal health laboratory facilities. With this, I would like to thank you for this opportunity. Thank you.